Welcome back, friends. In this video, we'll be talking about metabolomics. We have discussed about proteomics and we know what is genomics. We have studied the several different part of proteomics and genomics in detail. Now, in this video, we'll be talking about metabolomics and this PPT, actually this PowerPoint presentation, is most of the PowerPoint presentation is was taken from David Wishart, which is a very magnificent PowerPoint presentation for the metabolomics, which it's also a combination of other but most of the slides are taken from this person, so total privilege goes to him. Anyways, let's begin. Okay, now the first thing is the definitions and background of metabolomics. Now I've already discussed about it in the proteomics discussion, that we need to understand central dogma of biology very well. So we are having DNA, from the DNA RNA, then from RNA we are having proteins. Now in each level, transcription and translation, in each different levels there can be variations. Now those variations give rise to, as we are going from DNA towards proteins, the variation is increasing throughout the place. Okay, now in this case of metabolomics, we are not talking about up to protein level, we are talking about furthermore. Once we know that each cell contains many different metabolites, many different molecules, like chemical molecules which are generated during different cellular processes, and we all know that the different processes of metabolism like glycolysis, Krebs cycle, which are the major processes, then the protein metabolic processes, different metabolic pathways are fed onto several different types of molecules. Now all those molecules which are chemical molecules are called metabolites, and we all know that, right? So metabolites are chemical components that are found vigorously inside a cell. Many different metabolites are found. Now all those metabolites are part of different type of metabolic pathways, either that can be catabolic pathway or that can be of anabolic pathway, but they are having metabolic, they are coming from metabolic pathways. Now in those kind of study, when you talk about all those metabolites that are present inside a cell at a particular time, we call them metabolome. Okay, so it's similar like the definition of proteome. So all the metabolites, whether it is a primary or secondary, coming from the metabolic pathways which are present or found in on a particular cell at a particular given time, will be called as metabolome, right? Now, the study of all the metabolome inside the cell is called metabolomics. Okay. Now, as we are coming from the DNA to proteins, the information we're talking about, it is decreasing because the proteins are having less, and so what we are having, sorry, the information is getting increased. The information is increased because DNA is having only 4 base, RNA is having also that same type. Proteins are also having 20 amino acid, but metabolites are many more, right? So information is increased and the variation is also increased, but the material is getting decreased because we are having less amount of this, right? Okay, now so this is again another important thing, that what is the hierarchy of our study is that first of all we need to predict what is possible inside a cell. Now the second thing is the current direction of the cell. So what appears to be happening inside the cell. Then what we think or hypothesize about that what is appeared to be happening, we need to look for what makes that happens. And then finally what is happening inside the cell and how it is happening. So these are the questions we need to get the answers for, right? Now if we look at the metabolics in detail, so here it is the genomics. 40,000 genes, remember. Then we are talking about transcriptomics. We are having 1,50,000 transcripts. Then we go to proteomics. We are having more than 1 lakh. It's above 110 lakh of different types of proteins, right? Then finally, when we are talking about metabolites, there are 2,500 metabolites. So less amount of all. So metabolomes are present in the least amount inside the cell among compared to genomics, transcriptomics, or proteomics. Okay, and here in this data in this particular slide, it is adapted from this particular section, so reference is given or mentioned here. And this is important because it will tell you many informations about things like the specimens we take for this different purposes are also given. For the metabolomics, what we take biofluids, tissues, as well as cells, so we get metabolites from them. And the techniques for detecting metabolites are the NMR spectroscopy, GCMS or gas chromatography, mass spectrometry, and obviously mass spectrometry is an important part. We have talked about proteomics, where you have seen we are talking about mass spectrometry, 2D gel electrophoresis as the major applications or major tools for analyzing proteomics. But for analyzing metabolomics, we are relying vastly or majorly on mass spectrometry, combined mass spectrometry like GCMS or like NMR spectroscopy and crystallographic techniques. So what we are going to do, what we want to do is a prediction of life processes, right? What we want to find is the functionality of a cell or functional behavior of a cell. And to get this answer, we need to really rely on genomics to study the whole gene, proteomics studying the whole protein, and now metabolomics are studying all the metabolome that is present in a cell, right? Because we always want to know what is life, and to understand life we want to know how the cell functions, right? So here it is the DNA, which is the storage material of all information. It transcribed into RNA, then finally it is translated into proteins. Now here this part is called the gene expression part, which we can detect using proteomics. But now after the proteins, these proteins are taken for different biochemical pathways, which are also called metabolic pathways. It could be of catabolic uh, or degenerative and anabolic or constructive pathways. Okay, now these biochemical pathways give rise to different type of phenotypic traits. Like here in this case, the color of this flower is blue, yellow, and this is a one example of how these biochemical pathways are related and how they are related to phenotypic expressions or traits, right? So we can only see these traits by looking into the different organisms. Now we need to detect everything else inside this organism to find out what is triggering this trait, what is triggering the change in this color of the flower, for example in this case. Okay, so this latter part from the protein, which is involvement in the biochemical pathways, are also dealing with different type of metabolomes, and we are talking about here comparing all those metabolomes, how they are interacting, and how they interact in the normal cell, how they interact in the diseased cell. So we can do all these things by simply getting a map of all the metabolites and how they are interacting with each other. So this is our actual goal, is to produce or provide a map or provide a framework of how the metabolites are getting formed from different interactions, how they are getting produced, and how they are interacting with one another. Okay, so this is our goal using metabolomics. So here it is, so different pathways, and we can simulate those pathways via the computer analysis like bioinformatics tools like SwissProt, GeneBank analysis, and all these things. And using this simulation, we can also analyze the protein and metabolic profiles, and also we can analyze the gene expression. Now, if we look at the pyramid of life, we can see the environmental influence can be increased as we going towards the from genes to this metabolomics, right? So in the gene, the environmental influence is less. As we are going towards protein, due to different variations, the variation are increased due to different kinds of processes like post-translational modifications, post-transcriptional modifications, and all these things. So finally, in metabolomics, there are different types of metabolite interactions, different pathways in different orientation. For example, if oxygen is present, the metabolic pathway changes. If oxygen is absent, the metabolic pathway changes from the normal. So these things, simply the change of oxygen can trigger different types of metabolites inside the cell. Simple change in temperature and variations in light intensity and all these things for a plant will vary all the metabolites that the plant is providing or producing, right? So these things are important because the environmental influence is maximum to these metabolites. So we've talked about metabolomics earlier, so we don't need to talk about it now. And what is metabolite? So this is in definition you must know that any organic molecule detectable in the body with a molecular weight of less than 1500 Dalton. It means 1.5 kilodalton, or we can write it 1500 Dalton, right? So any organic molecule detectable in the body with a molecular weight of less than 1500 Dalton will be called as metabolites. Now the example of metabolites are peptides, oligonucleotides, sugars, nucleotides, organic acids, ketones, aldehydes, amino acids, lipids, steroids. You can see most of the things are among the part of these metabolites. Like, and obviously drug and drug metabolites. That's where we come to know this metabolomics very very important, because we need to learn the drug and the mechanism of drug, and how the drug is targeting different diseases, and how we can manipulate this using metabolite or metabolomic techniques. Okay. Okay. Now, here are the different metabolomes. We don't need to talk about that here. Okay. Now, if we look at the human metabolomes, what we can find is that the endogenous metabolites are the maximum one. It is the first place, and then we are having drugs. Then we also have food additives, which is a part. So the metabolomes that we're talking about, it can be of different types. The complete collection of, so the metabolomes means actually the complete collection of small molecules, which are called metabolites inside the cell or organ or tissue or organism, whatever. Okay, but what we talk about, it could be a part of endogenous or it could be a part of exogenous. Now the endogenous metabolites are the maximum again, right? So this is the concentration variation. So this part is for the detection. So as we are going towards this part, it is the highest. As we are going from this part, it is the lowest one, right? So this is towards the highest one. Okay. Now here it comes, the endogenous metabolites are present in the highest concentration, but then we are having drugs. You can see inside the human body the drug is present as a common metabolites which is present in the second highest concentration. So you can take the higher drug uptake. So drug uptake in human body is increased how far? You can get this value by just looking at this image. And now also we are having food additives. You can see this is the food additive which is the second highest concentration of the metabolites that are present. Then the drug metabolites, and then finally the toxin are present in very few amounts in normal body. Okay. Now due to any kind of change, due to any kind of disease, suppose you need to take a lot of drug, then this profiling, then this metabolomic profile will vary. Now this is what is called a metabolomic profiling. Now the profiling means it means that inside a human body, a normal human body, what are the different concentration of different metabolites in a tissue. For example, it's if it is tissue specific, then take it's a suppose it's a liver tissue or liver cell. You take a liver cell and look for this thing. Suppose so in the liver cell, the metabolic profile is that the metabolomics, the endogenous metabolites are present in highest concentration. Then the second highest will be the drugs, third highest will be the food additives, fourth highest will be the drug metabolites, and the fifth one and the least concentration carrying will be the toxins, right? So these are the variations, right? So this is the profiling. Now if due to any kind of disease, this profile will be hampered. How? This kind of disease, suppose the endogenous metabolite concentration falls, and due to take off higher drugs, this concentration increases. So as a result of that, by looking at that, the endogenous metabolite concentration falls compare it to it the normal profiling, we can tell that yes, the organism is diseased by just examining the liver tissue from that diseased individual, right? Or suppose due to any kind of bacterial infection, the toxins or environmental chemical assumption concentration increases inside a body of a particular organism, of body of a suppose a human being. Now if we take him and take the liver tissue of that particular infected individual, and we make this metabolomic profile and compare this metabolomic profile with the normal metabolomic profile, what we can get, that we can see this toxin levels are increased. So this type of indications will tell us that there is something wrong about this particular diseased individual, and it help us to detect what type of disease the person is having, right? So this is the theoretical metabolomes mean we are talking about the secondary metabolomes, right? Now, what are secondary metabolites? We all know that primary metabolites are those metabolites which are produced during the very basic pathways of metabolites or metabolic pathways. But after those metabolic pathways, what we have generated, say the pyruvate is generated from glycolysis and all those different things. Now, if we take those pyruvates and we produce something else, suppose we produce an amino acid with that using that pyruvate. So this pathway is not direct one. This is not the primary pathway. So the product of the secondary pathway will be called the secondary metabolite. So suppose here it is a glycolysis, we produce this pyruvate, but this pyruvate, so this is the primary pathway. So this is called the primary metabolite, this pyruvate. Now if we take this pyruvate for the future purpose, suppose this pyruvate is taken to synthesize an amino acid. Now this is not a primary pathway, biochemical pathway. This is a secondary biochemical pathway. So we call this metabolite, this particular amino acid derivative, as secondary metabolites. Okay, so like that. So during the secondary metabolite also, the more or less this metabolite variations remains the same, because the secondary endogenous metabolites are the highest concentration, then the secondary food metabolites, then secondary drug, and then the lipid and lipid derivatives are present. Okay, so these are the two different types of metabolomic profile. So we need to provide this, or we need to create this metabolomic profile of each different tissues inside our human body. So if we can provide that, then it will be super easy for us to understand a disease, or to understand the onset of a disease by simply comparing the profiling of this metabolomic with the healthy person and the diseased person. Okay. Now, factors influencing the metabolome. So there are several factors that influence the metabolome, right? So this is another important point. So unlike the proteomes are also important, or also influenced by different metabolomes. But most of the case, as we are going from the DNA towards this metabolomes, the influencing nature of the nature is getting increased. We have already seen that. So what kind of influence that we can get? It depends on your diet, your gender, environment you live in, depends on your age, then genetics, and obviously drug treatments, hormones, lifestyle, and all these things are involved, right? So everything here is involved, right? So we cannot detect it by simply looking at this metabolomic profile. So your metabolomic profile will be so this is the problem with this metabolomics. Remember, I've told you that we need to provide a standard, what is called a standard metabolomic profile, right? To produce the standard metabolomic profile, we have seen that these are the standard metabolomic profile of a human being, normal human being. Now we have also seen that this can be varied from person to person, because it varies from gender to gender, from diet to diet, from the lifestyle, or the variation of age. So that means it is extremely hard to produce what is called a standard metabolomic profile. We cannot produce a standard, because what is meant to be standard for me will be varying for you, will not be the standard for me, because my lifestyle is different. Because I'm an Indian person, my lifestyle is completely different for a person who live in US or UK. And obviously in the Western world, the culture, the lifestyle, the diet is completely different. So definitely their metabolic profile will be different than us. So if we get the standard metabolic profile built for an Asian, that will not compete with it with the European. So these are the examples, these are the variations, right? So it creates extremely hard to provide this metabolomic profile. But still the research is going on to in search for a suitable metabolomic profile. Okay, now why metabolomics is important? It is important because it is the study of all metabolites, and we have seen why we are actually talking about metabolomics, because it will help us to identify diseases, and also it will help us to identify and look for what is a suitable and perfect metabolomic profile. Okay, and we also know metabolites are small molecules, and we are also getting some information about the small molecules. And we know that greater than 90% of the diagnostic clinical assays tests are for the small molecules, and obviously 90% of all known drugs are small. So we are getting informations about those small molecules in our body. So it will help us to understand the drug targeting and obviously different disease conditions. And metabolome is connected to any other type of ohms. Ohms means suppose genomics and obviously proteomics. It is connected everywhere, because the proteins that are provided from the proteome is also a part of the metabolomics. Okay. Now the metabolomics enable the system biology to understand what is we are talking about, right? So here it helps us to build what is called the system biology. Systems biology means we are dealing with genomics, dealing with proteomics and metabolomics, all of them together, right? So it helps us, provide us one section of the uh, what we can call it a systems biology. Okay, now the application of metabolomics for many purposes. So you can memorize some of them. Major applications are toxicology testings, clinical trial testings, and obviously food beverage testings, and drug phenotyping, water quality testing. Then obviously obviously what you can say, genetic disease testings here, and nutritional analysis, drug compliances, cholesterol testings, clinical blood analysis, and all these things will be part of the application of metabolomics. The metabolomics methods are we can see the environmental influence is increasing, right? So let's look at the metabolomics methods here. So this is the workflow of how metabolomics work. So here suppose this is the biological tissue or sample, whatever we are taking. So it could be bacteria, it could be eukaryotes like a animal or plant, whatever we take a tissue, and we homogenize the tissue, we get our extract from the tissue. And then when we get the extract, which is called the biofluids or extracts, we take them and we look for the chemical analysis. Now the chemical analysis can be done using NMR spectroscopy, it can be done using the HPLC, it is the HPLC, or it can be analysis by the gas chromatography. And then finally we need to identify what are the molecules or what are the metabolites that are present there, using what is called MS or mass spectrometry. We can combine this a different technique like gas chromatography with mass spectrometry and other things, finally get a better result and better resolution of what are we dealing with, right? Now, why metabolomic is difficult? Because the chemical diversity is extremely increasing during this process, and obviously, it is hard to construct a metabolomic profile due to because it is the environmental factor greatly influence the metabolite presentation inside our body, right? Now, the technologies that are usually used for metabolomics are UPLC, HPLC, obviously LCMS, which is a type of mass spec, FTMS another type of mass spec, NMR spectrometry, X-ray crystallography, GCMS, and LIF detection. So these are the different techniques. You can see HPLC, you can see NMR, you can see here the mass spec, and obviously different techniques. Okay. So what is possible using this? So NMR based metabolomics is a part, GCMS based. So these are the different based metabolomics part, and most of them are utilized for identification of the sensitivity. You can see the sensitivity level can be increased using the lipomics and obviously LCMS based metabolomics analysis. So there are actually two routes of metabolomics. One is the quantitative, which is called the targeted method. Another one is called the chemometric or the profiling method, right? So let's look at it here. So for the profiling method or profiling method or chemometric method of our discussion, what we can take the sample preparation, then we run the we extract this sample, whatever we get the sample, and then we uh, feed this sample onto the analysis program like LCMS, HPLC, whatever we get the data, and once we get the data will be reducted. The data reduction will be done using some control values, right? So using some control values, you get the value of the data. You can interpret what this data is meant by comparing it to the control value, and we finally get the metabolite identification. Now we can rely upon different bioinformatics servers for the detection of this metabolomics and all these things. It will be a part of it. And second process is the quantitative or targeted. Now for this process, what we need to do, we need to take the sample preparation. Then what we do, we are having a metabolomic identification and quantification, and then we have data reduction, and then finally what we do, a biological interpretation of how they are interacting. Remember, this is called the quantitative. This is a much difficult one, right? So the first type of technique is a profiling. We need to profile what type of metabolite we're talking about. So we first profile them, we understand what metabolites we're dealing with. After knowing that, what we do, we need to feed this data onto the software programs, algorithms, so that it will help us to interpret how all these metabolites are interacting with each other via different biochemical pathways, so that we can get a clear picture or clear understanding of how metabolic pathways are linked with each other using these metabolites. Okay. Now once we get our spectra, once we get our this desired biological interpretation of our identification of the metabolites, then what we do, we produce a list by using this spectra, and from this list, what we get towards the pathway. So that's how we derive the metabolic pathway and how all these metabolites are linked inside the pathway. So this is how the technique is used for the metabolic pathways. Remember, we have talked about several pathways like lipid metabolism, carbohydrate metabolism, amino acid metabolism in many different parts, but we didn't discuss that how we come to know about all these things. We come to know using this metabolomics, right? So we use these technologies, and then derive how they're interacting. We provide the list, then from the list we provide this metabolic pathway. Okay. So you can see here, by simply comparing it, by looking at them using the chromatographic techniques as well as using the mass spectrometry, what we get is what is called the compound. We identify the compound using mass spec, and also using GC or gas chromatography or high performance liquid chromatography, what we get, which is called the retention time. Remember what is retention time? If you don't know what is retention time, please go back to my chromatography videos and you will find it. Now once we get this retention time, that means during the process of metabolite pathways, which product is coming fast, which product is coming late. So we make a list of this compound with their retention time from low to high, and once we get them, we can get the understanding of which of this metabolite come earlier in the metabolic pathway, which is coming later. Then what we can draw this kind of metabolic pathway, right? That's how we get the pathway from a beginning of a cell and tissue sample. We go towards this pathway. So what we can do now, from these pathways we can make models of biomarkers. So this is much more further. This is a very new type of work. We can model it in a virtual world. If we are modeling this virtual world, what the cell will look like and how the cell will function in this virtual world. Okay. to understand finally how a cell is working and how life is working, to answer the question what is life. So this is the basic, at the end we're going to answer these questions, right? So this is the application of biomarker identification, and for a disease we usually use this for the biomarker application. I don't want to talk about that, but this is how we come to the end using this metabolomics. Okay, so that's it, and I hope that's helpful. Thank you.